Okay, middle lamella, you're going to see only in plant cells. Holding a plant cell to another plant cell. And that's, I'm going to skip a little bit. Plasmodesmata, okay? And that's why I brought the plant cell, okay? And the middle lamella, okay? The plasmodesmata, and the middle lamella. Okay? Okay, these would be the plasmodesmata. This, again, would be the entire cell wall of the individual plant cell that this model is. This would be the wall of this cell. This would be the wall of another cell that you don't see here. So the middle lamella is the structure holding, I refer to it as the cement holding one or more of these plant cells together. Okay? So if I put a label here, okay, and ask for the fibrous structure that's labeled in this plant cell, you would say the middle lamella. Okay? Or if I put a label here, you would say, and again, ask you, identify the labeled structure. You would say the plasmodesmata. Okay? And again, holding, these are open gap junctions. So nutrients and metabolic waste product from this cell can go from this cell to this cell here that you don't see. Just the cell wall. It has to go through middle lamella, diffuse through, and then open gap junction or another plasmodesmata, and then where? Another plasma membrane of another cell. Again, there's a plasma membrane almost adhere to this plant wall. You don't see that. Okay? Every single cell has a plasma membrane. That butter sandwich, including plant cells. But that turgor pressure that I was talking about holds that pressure almost adhered to the cell wall would be the plasma membrane of this plant cell. Again, you'll see just a cell wall. Some students will just say cell wall and forget about plasma membrane. And that's why tomorrow I'm going to have you do the same thing, look through Elodea, make a wet mount of an Elodea leaf, and look at individual healthy Elodea cells that you already saw yesterday. And then I'm gonna have you add a little bit of salt. And then see the plasma membrane ripping apart from the cell wall. And I'll show you that momentarily. But again, the plasma would be these gap junctions. So again, the plasma membrane will determine what goes into and out of this cell. Even it'll get stopped here with this gap junction, and then be determined by the plasma membrane of this cell. And again, metabolic waste products can go through and diffuse through the middle lamella into another plasma desmata. Oh, and yeah, you're gonna see this vesicle. And again, the vesicle up here, you're not going to see a membrane around a vacuole, right? But you will see a membrane around a vesicle. Okay? So again, vesicles are produced by the Golgi body. They are membrane-bound vacuoles. It's a good way to remember. A vacuole is just an air spot within the cell, usually used for storage. Okay? But my point here is that vacuoles will never have a membrane. If you see a membrane around a vacuole, it's not a vacuole, it's a vesicle. Okay? That you see here. Okay? You can see a little ridge representing a membrane. And again, another one here. Again, these are used for exocytosis. Okay, that I just went over. Okay, I think I'm all set. Right. There's three main animal cell junctions. Like the extracellular matrix, the middle lamella I mentioned, animal cells. We'll see three different types of junctions in animal cells. Tight junctions, adherent junctions, and gap junctions. A gap junction is, again, open cell. So wherever that gradient, high to low gradient, less energy, okay, going through a gap junction, as the name implies, it is just one gap. Okay? Free flowing, okay? Working on concentration gradient. Okay? Like I just pointed out with the plasma desmata, every single plasma desmodium is a gap junction. Adherent junction. We'll see this in the integumentary system of mammals. The skin, okay, easily stretch apart. Okay. There, and I have images depicting all of these. Whereas the seal, or the tight junction, okay, almost like spot welding. Very, very. This is a tight junction, or what I refer to as spot welding. If you've ever seen or done any welding yourself, you can definitely see the rivets, very, very holding tightly together. One cell, one plasma membrane to another plasma membrane. And again, this would represent one cell, and this would represent another cell, and this would be a third cell. The gap junctions are free flowing. And then the third type would be the adherent junction. And these would be the microtubules, would be microtubular lattice, would from one cell would bleed into another cell. So again, whatever communication is being communicated to by this cell, it's easily transferred to this other cell. We'll see all three of these in the histology portion. The plasmodesmata I mentioned. This would represent the, this is a artist or a cartoon based on a micrograph taken from an electron microscope. This would be the wall of one plant cell, right here, intracellular, and this would be the wall of another plant cell. And this cementing, if you will, would be the holding the two, known as the middle lamella. Only in plants would you see plasmodesmata and middle lamella. This is a little table I put together. Plant cells, the cell wall. No animal cell will ever have a cell wall. Yes, no animal cell. Plant cells will have, every single plant cell will have, most, if not every single. Cellular plasma membrane or cell membrane, that butter sandwich, if you will, or the bilipid layer, both will have the same. Chloroplast, no chloroplast, and yes. Again, in order to be a member of Kingdom Planta, you need to photosynthesize. So you need chloroplast. Centrioles, no. Flagella and cilia. Only in plant cells. No animal cells. You didn't hear about the fern gamete. But they have
Flagella. Extra credit question. Bring it out there.
Yeah. Okay. Mitochondria. Yes and yes. Oh, wait a minute. Wait a minute. Wait a minute. Photosynthesis. You're taking in carbon dioxide, and one of the end products of photosynthesis is oxygen. So plants give out oxygen, and they take in carbon dioxide. We do the opposite. We take in oxygen and expel carbon dioxide. But wait a minute. I just said plant cells have mitochondria. So therefore you should be thinking, what? The same thing I'm thinking. Plant cells need oxygen. I just said, oxygen, without oxygen, yes, aerobic or anaerobic respiration will occur. You're only producing 2 ATPs for every glucose broken down. But with the mitochondria, you're able to produce up to 36 ATPs. So therefore, with aerobic respiration, aerobic needs oxygen. So plant cells, in essence, need oxygen. So keep that in mind. Okay? They expel most of their oxygen, but they have to keep some of their oxygen. If they're using the mitochondria, and yes, they will have mitochondria, and yes, they will aerobically respire. Keep that in mind as well. Eukaryotic versus prokaryotic. Don't want to belabor the point. You see some sleepy faces.
Can you just touch on the nuclear envelope and the ribosomes, though?
Okay. Yes. Large ribosomes and the small bacteria and all prokaryotic cells will have much smaller ribosomes than eukaryotic cells. And the nuclear envelope, I mentioned, I don't like to refer to that as the nuclear membrane, and it's referred to as the nuclear envelope, simply because if you're thinking of a membrane, you're thinking in a biological term, you're thinking of the phospholipid bilayer. Okay? But the nuclear envelope does not have that bilipid layer. And obviously, without a nuclear envelope or a nucleus itself with the prokaryotic cells, you're not going to have to worry about the nuclear envelope in the prokaryotes. We'll talk a little bit about these. There's 2 parts of a ribosome, a small subunit and a large subunit in eukaryotic cells. A small ribosome in prokaryotic cells. We're going to talk about that later on with the molecular genetics. Okay, the plasma membrane, the function and the structure, the fluid mosaic model. This refers to what I showed you, and I stopped in that video. And my analogy of a walking through into a crowded elevator. You're weaving around. Without those cholesterol molecules, that fluidity is not there. Okay? So again, the fluid mosaic model. Mosaic is little bits and pieces of it. Think of the word mosaic. You're producing a large portrait by little bits and pieces. So the little bits and pieces would be the individual phospholipids. The fluid would refer to the fluidity of the membrane. How they individually, phospholipids, jockey for position. And that's where originally, when the electron the biologists back then, they just thought that the membrane was like a piece of saran wrap holding the organelles in place. It wasn't until the electron microscope that biologists were able to see the amazing structure of, and whatever. It was always thought that the nucleus will control the activity of every single cell. Well, the nucleus is within the cell itself. And how can the nucleus inside the cell sense the outside environment, the temperature, the conditions, the light or dark? Okay? But again, with the electron microscope, you're able to see these molecular antennas that will sense the outside environment and transfer that information to the nucleus. So again, the nucleus is not controlling the cell, so to speak. It's the membrane, outer membrane itself. These molecular antennas, I like to refer to them. Again, the main function is to allow nutrients coming in and hormones, protein-based hormones, any advantageous material to enter the cell, and metabolic waste products to exit the cell. And that's done by 3 different types of cells. I may have you, again, another short answer or 5-point essay, short answer, to draw typical plasma membrane and describe typical plasma membrane. But again, I don't want something like this here would be good enough. And then so what I'm looking for are these carboxyl heads and 2 fatty acid tails representing one phospholipid. And again, the arrangement like this here. Okay. Another set of phospholipids, tail first and then head. And this would be intracellular, and this would be extracellular. And then these would be the proteins, or the molecular gates. So materials could enter and or exit through these molecular gates. And you can do a very elaborate, if you're molecular antennas, receptor molecules. But anyways, something like this would be worth a few points. And then explain what is the biological control or the important biological importance of the plasma membrane. This would represent, again, these would represent the membranes, and these too. Okay? So again, whenever you see this phospholipids, heads, tails, heads, individual, and then these would be the molecular gates made up of the proteins. Transfer proteins. Okay? You could have a gap junction. Okay? This would represent open gap. Okay? This would open pore. Okay? Again, there's a higher gradient on the outside. So you can see these atoms or molecules, there's more on the outside, so they'll flood in. Okay? There's only 2. So when 2 more come in, they'll equalize and be no movement in. You need that gradient, okay? A high to low gradient. With the energy or the you need some type of energy to open this gate and then shut it. Okay? And then another type of energy to open it from the inside. Okay? So that energy within the cell, you need ATP. Okay? So again, embed it within the plasma membrane. Receptor recognition proteins, intercellular interaction, or cell 2. You can see the plasma membrane of this cell. Okay? And this would be the plasma membrane of another cell. Okay? So you know interaction coming together. These may be leukocytes or white blood cells, right? Moving around. When they see a target cell, right, it, the same shapes or the same, they'll stop right here and they'll lock on, right, and change the metabolism of the what's going on within this other cell. And then finally, receptor proteins. Binding hormones, primarily the protein-based hormones. Hormones. Remember the fat-soluble hormones. Most hormones are fat-soluble or steroids would be easily, but some hormones would not be easily. Those would be the protein-based hormones. And again, a hormone is technically defined as any biological structure formed in one or more cells within that organism and then transported throughout that organism to what is known as a target cell. So technically, this is what a hormone is. Produced in another cell and then transported throughout that organism till it finds a target cell. And those individual target cells will have receptor molecules specifically to bind like a lock and a key. Not every single lock will unlock every single key, or vice versa. Every single key will not unlock every single lock, except for specifically shapes. And that's the molecular, different shapes of molecules. And we saw with the, I pointed out, the difference between cholesterol, estrogen, and testosterone. One hydrogen and testosterone, and it's a hydroxide, okay, OH.